Hello, everybody, and welcome to chapter 11. We are going to talk about the blood in this chapter. This is our first chapter of Unit 4. And so, Unit 4 is all about the blood, the heart, our circulatory system, and then we finish up with the lymphatic and immune system, which is very much related to our circulatory system. But this is going to be a good background into blood itself. So our blood has many functions. If you remember back from our previous chapters, blood falls under the connective tissue category, and it's unique because it's the only liquid connective tissue. So its extracellular matrix is liquid. We call it plasma. And because it is liquid, it allows it to serve a large variety of functions, and it acts somewhat as a highway for transportation of materials. So you'll see our first three functions here all relate to transportation. So these are all things that we can find within our blood, and our blood is a good way to get them to where they need to go in the body. So we have gases, nutrients, and waste. So stuff like oxygen, carbon dioxide, glucose. So all the stuff that our cells need to function and the waste from their metabolism. Processed molecules. So whether it's enzymes, proteins, steroids, any other molecules that are made within the body that may need to be transported to another area. Regulatory molecules. We just talked in chapter 10 about the endocrine system. And so think about hormones traveling through the bloodstream as their method of transportation to their target tissues. Our blood helps us maintain homeostasis with regulation of pH, or our acid-base balance. It also has a role in osmosis. Remember, this is the movement of water, and so this happens between the blood and our body's tissues. There's water movement to help regulate and maintain proper concentrations and pressures. It helps us with body temperature, so we'll talk about that a little bit more in our future chapter on the circulatory system. But our blood vessels, namely our arteries, are able to dilate and constrict to allow heat to be released or maintained within the body. Protection against foreign substances. So we'll talk about some white blood cells that are involved in our immune system, and we will also talk about clot formation. So the actual makeup of our blood, plasma is the fluid portion, and this is a majority of the blood, about 55%. It's a pale yellow liquid. It's mostly water, about 91% water, but there's also proteins and then some other dissolved substances within the plasma as well. We then have our formed elements. So these are going to be our actual cells and cell fragments that make up the other 45% of our total blood. And these are composed of erythrocytes, which are red blood cells, leukocytes are white blood cells, and then we have our thrombocytes, which are also known as platelets. So these are the primary plasma proteins that you guys need to know. So remember, plasma is that fluid portion, and these proteins are contained within the fluid. So albumin is the majority of our plasma proteins, and its main job is to help maintain water balance. And so the movement of albumin will draw water with it. So that's how we maintain our water balance. Globulins are a class of plasma proteins that make up another large portion. These are involved in our immune system response. And then we have fibrinogen, which is about 4% of plasma proteins. So fibrinogen, when it is released, is activated into fibrin. And so fibrin is what aids in clot formation. So we'll talk about that on a slide a bit further in today's lecture. This is just a good summary slide here. So you see, if we were to spin a blood sample to get it to separate, we call that the hematocrit. And so the plasma is on top. We have this buffy coat, which is some of our white blood cells and platelets. Then you have your formed elements, which are made up of mostly red blood cells. And this just shows a little bit more specific of a breakdown. But like I said, this is kind of a good summary slide for what our blood components are. Okay, erythrocytes are our red blood cells. And so they are this biconcave disc shape. So you see they're a circular disc and they have a little indentation on both the top and the bottom. So that's why it's called a biconcave disc. You'll see they do not have a nucleus. So think to yourself for a second, what would that prevent them from doing if they don't have a nucleus? So if we don't have a nucleus in our cell, we don't have that cell's DNA or instructions for replication, which means our red blood cells are unable to replicate. They only live for about 120 days and then they're recycled, kind of broken down into their component parts. Our main job of the red blood cells or our erythrocytes is to transport oxygen to our tissues. And we rely on a molecule known as hemoglobin to be able to transport oxygen. So this is the main component of our erythrocyte. It's how we transport oxygen. So hemoglobin is named for its two components. We have a globin protein and a heme molecule. So heme plus a globin equals our hemoglobin. And within our heme molecule, there's an iron that oxygen is able to bind to. All right, so each red blood cell is going to contain 4 hemoglobin molecules, meaning that each red blood cell can carry 4 oxygen molecules. So one red blood cell can carry 4 oxygens or O2s. And so when our hemoglobin and red blood cell actually has oxygen attached to it, then we refer to it as oxyhemoglobin. So oxyhemoglobin is when we have oxygen attached to that hemoglobin on our red blood cell. And so if you've ever been to the doctor and they put the little clip on your finger that measures the amount of oxygen in your bloodstream, what it's trying to kind of look at is how saturated or how much oxyhemoglobin do you have compared to your total amount of hemoglobin in red blood cells circulating. So a normal healthy person with no breathing issues is going to be at 98, 99% oxygenated. So this is just the hemoglobin molecule. Like I said, it's made up of 4 globin proteins. Each have a heme molecule. So that's hemoglobin. So one red blood cell can carry 4 oxygens because each heme has an iron for bonding. All right, leukocytes are white blood cells. So when you think white blood cells, you should primarily think immune system. So their main job is going to be fighting infections and foreign invaders into the body. And they'll also remove our own cells that are not functioning properly or have died for whatever reason. And so these are going to be a good bit larger than an erythrocyte under the microscope. And these do contain a nucleus. So these can replicate. So we classify our leukocytes into a few different categories, the first being granulocytes versus agranulocytes. And so our granulocytes basically mean cells with granules. So these have specific granules within the cells. So our 3 main ones are neutrophils, eosinophils, and basophils. So our neutrophils are our most common. And so these are going to be in our bloodstream for 10 to 12 hours prior to migrating actually into the body's tissues to stay. And these are phagocytes. So remember, phagocyte is a cell that eats other cells. So our neutrophils are going to be the most common, probably about 80% of our white blood cells, and they're going to eat up foreign material and dead cells and debris. Our eosinophils are associated with reducing inflammation. And if we were to have a parasitic infection, our eosinophils would respond to that as well. Our basophils are our least common type of granulocyte. We don't have a lot of them, and they're associated with releasing histamine and heparin. And so we'll talk about that a little bit more in our following chapters. There's also agranulocytes, meaning they don't have any specific granules. And we have our monocytes. These are large white blood cells, and they produce macrophages. So macrophages are matured monocytes. And so these are also similar to neutrophils, going to be a phagocytic cell. They're going to eat up foreign material, dead cells, debris. And so they will migrate into the body's tissues eventually as well. So they'll circulate the blood in the bloodstream, and then they'll migrate into a tissue. Our lymphocytes, we'll talk about this mostly in chapter 14, but these are involved in our specific immune response. So we have two main lymphocytes. We have our T cells and our B cells. We're not going to really get into it much more in depth than that right now, but they produce antibodies and they respond to our antibody-mediated immune system. So here's what our white blood cells look like under a microscope. You'll see not an amazing picture, but there's little granules in our granulocytes, and our lymphocytes and macrophage or monocyte just have the large nucleus there. So these granules stain different colors under a microscope, so we can tell which type of granulocyte it is. All right, moving on to platelets. When you think of platelets, you want to think of blood clotting. So they have a really important role in preventing blood loss. We call them platelets because they are actually fragments of a giant cell known as a megakaryocyte. And so our megakaryocytes are giant cells, and these little pieces break off that become our platelets. And so these are produced in the red bone marrow, just like our red blood cells are. And so we're going to talk about clotting here a little bit. So when blood vessels are damaged, blood is going to leak into other tissues where it's not supposed to be. So that can definitely disrupt our normal function. And if we lose enough blood, we have to replace that either by the production of new blood ourselves, known as hematopoiesis, or by transfusion, which is where you would say go to a hospital or somewhere that you would get a bag of blood transfused into your body. And we'll talk about that process at the end of this lecture. In terms of preventing blood loss when damage does happen, we have a few steps. So the first step is vascular spasm. So when a vessel is initially injured, it is going to spasm or contract to try to constrict the amount of blood flow that is going through that area, and therefore the amount of blood lost. So I always say, think of if you got a cut on your arm or wherever, the first instinct is to like grab a towel or your hand and put your hand right on top of that cut to try to hold it down to prevent blood flowing. That's kind of what your artery is trying to do by constricting and squeezing its smooth muscle layer to slow down the blood flow. Once that area is damaged and there's exposed vessel wall, platelets, as they float by, are going to be able to stick to that damaged area, and they're going to stick to each other, and that is eventually going to form a platelet plug. And so this is kind of like putting a cork in a bottle. So it's going to seal it up some, but you're not going to be able to kind of seal it up 100%. It's a good temporary hold. And eventually a full clot is going to form. And so our blood goes from somewhat of a liquid state to a gel state, and that acts as a glue in a sense to hold our vessel together before it can fully heal. So clots are great when we need them, but we don't want them there forever, so we need to be able to get rid of them. So clot retraction is the term for getting rid or condensing a clot. And so we just said our clot ends up kind of like a gel. And then we have serum within our plasma, which is kind of purely the water portion is going to be squeezed out of that clot. So our gel is going to have kind of the water squeezed out of it. It's going to dry it out, and that's going to help the actual healing process. And we also go through a process known as fibrinolysis, and this is how we dissolve the clot the rest of the way. So earlier on, we talked about the protein fibrinogen, and I said that when fibrinogen is activated, we have fibrin. Our fibrin acts somewhat like thread or a spider web to attract platelets and help repair our injury. When we want to get rid of that fibrin, we then activate plasminogen, which is another plasma protein that ends up activating into plasmin, which breaks down our fibrin. So we get rid of that clot. All right, we're going to talk about blood grouping. And so this is an interesting concept, but it can be a little bit confusing. So try to follow along with me, rewind if you need to, and I'll try my best to make it as clear as possible. So we said if we had enough of a blood loss, we may have to get a blood transfusion. And so with blood transfusions, they have to match your blood type, where they have to give you blood that your body is going to be okay with. If they give you blood that your body is going to react to, then we can have a reaction known as agglutination. And so what that is a clumping of our red blood cells. More or less, our body starts attacking our red blood cells, and that's not a good thing because then it can't do all those functions that we listed at the start of the lecture. And so there's a reason we have these blood groups. So you may know your own blood type. You can have A positive, B positive, O, AB. So those are all types of blood. And then there's a positive or a negative tagged on to that based on what's known as an RH factor. And we'll talk about that in the second part. So we're going to focus on just the letter portion, kind of that A, B, AB, or O. And so a few terms that I need you to know to understand this is the term antigen. So antigens are going to be on the molecule or are a molecule that are going to be found on the surface of our red blood cells or the surface of our erythrocytes. So our antigens are on our erythrocytes. Our antibodies are proteins that are floating around in our plasma, and their job is to locate specific antigens. So every antigen has an antibody that matches up with it. And then our blood group, like I said, is going to be named according to the antigen. So you're going to have either A antigens or B antigens, or in the AB blood type, you'll see we have both antigens. And so we'll walk through this some. So I just said type A blood is going to have A antigens, type B blood is going to have B antigens, and type AB blood is going to have both antigens. Type O blood has neither. You can think of our type O, remember 0, so that's got no antigens on its surface. And the types of antigens that are going to be found on our surface are just genetically determined. We have no control over that aspect. And so O positive is the most common blood type among everyone. And we'll talk about blood donors here in a little bit. So certain blood types, like they have specific antigens, also have specific antibodies within their bloodstream. So if you took type A blood that has A antigens on its surface, they would also have anti-B antibodies. Type B blood, which is going to have type B antigens, is going to have anti-A antibodies. So if you were to put type B blood into a type A blood person, those anti-B antibodies would attack those new B blood cells because they contain the type B antigen, and vice versa if you put type A blood in a type B blood person. Type AB blood is not going to have either antibody because remember, it has both the A and B antigens found on the surface of its red blood cells. And then our type O blood actually has both anti-A and anti-B antibodies because it has no antigens on its surface. So here is a good summary slide. So if you can understand this slide here, then you are in good shape for the blood group and blood type aspect. So we're looking at type A blood in this first column here. So our top row shows you what the actual red blood cell or erythrocyte is going to look like. Our bottom row is showing what antibodies are present. So our type A blood has type A antigens. So our type A antigens are these little dots. So our type A blood has dots on the surface. Our type B blood has spikes on its surface. So type A antigens are the dots. Type B antigens are our spikes. Now, if we go back to our type A blood, you see we have anti-B antibodies that have this shape that fit into the spike. So if our type B blood was found in a type A person, then our anti-B antibody would lock on and cause that immune response. Similarly, if we had our type B blood, it has anti-A antibodies with a space to fit that dot. So if type A blood was introduced into a type B bloodstream, the anti-A antibodies would fit onto that antigen and you get a reaction. In our third column here, we have one of our special types. These is type AB, no antibodies at all, because both antigens, we have dots and spikes on our surface. And then we have type O, you see we have no antigens here, but we have both anti-A and anti-B antibodies. So type O are universal donors because they have no antigens. So they're universal donors. Anyone can get type O blood, because if we put type O red blood cells with no antigens in any of these other three types, there's going to be no problem because there's nothing to recognize as foreign. There's nothing on the system to fit into an anti-antibody. So type A can receive A and O. So of course it can receive type A, that's what they already have, and they can receive our universal, type O. Type B, same concept, they can receive B and O. So of course they can receive more B with their spikes, or they can receive O that has nothing on the surface. AB, you're lucky, you're the universal recipient. You can get AB, or O. There's no anti-A or anti-B antibodies, so no matter what they have on their surface, they have no problem with it. And then type O, even though you're a universal donor and everyone loves you, you can only receive type O blood because you have both anti-A and anti-B antibodies. So if you need to go through that one more time, kind of rewind and walk through it. Like I said, you should be able to understand this summary slide here, and these should make sense to you why they are accurate. So that was just the letter portion of our blood group. Now we're going to get into the positive and negative. So a full blood type is going to say, for example, A positive or B negative or O positive. So that positive or negative is based on this RH group in antigens. So RH positive means that you do have RH antigens in your system. So 85 to 95% of the population is RH positive. This is very common. And the only time it's problematic is when an RH negative person is exposed to RH positive blood. So this can happen two different ways. It can be introduced via a transfusion, or it can occur when mom and baby are two different RH groups. So we're going to walk through this example. So think mom is RH negative. So mom does not have RH antigens. she actually is going to end up developing anti-RH antibodies. So mom is RH negative, meaning she does not have that RH factor in her system. The fetus is RH positive. If any of the fetal blood leaks through the placenta during birth, it's going to mix with mom's blood. And so now RH positive blood is going to be introduced into mom's RH negative blood system. So the first time this happens, mom's system is going to say, hey, I don't know what this RH factor is. And it's going to produce antibodies to recognize and create an immune response against those RH antigens. So the first time it just realizes it happening and it tries to create a response in case it happens again. The second time is when it becomes a real problem because now mom's immune system is already ready to go. It already has those antibodies to recognize the RH antigens. And so if we get that mixing of blood again, then mom's system is going to kind of go on full attack, and that can be real problematic for the fetus. So it's called hemolytic disease. And so it's when those anti-RH antibodies end up crossing that placenta and attacking the RH antigens in the fetal system. And so now it's kind of a very known thing, especially with how high of a percentage of the population are RH positive. So in the case where you get that RH negative person, they just treat mom with something known as RhoGAM. And so it more or less neutralizes that reaction. So it becomes less of a concern. And so that is our last bit for blood. So we will continue on with chapter 12, which is our chapter on the heart.